I'm Nick Dirks. I'm the president and CEO of the New York Academy of Sciences. For those who don't know, it's been around for a long time, 207 years and counting. And we have done many things over those 2 centuries. We have the longest running scientific journal in America. And we have been hosting conferences for years and years and years, many of which are actually about new pharmaceutical products, new drugs. We did some of the first conferences in the mid-20th century on new antibiotics after the discovery of penicillin. And we've continued to be a place that is very interested in biomedical science in all kinds of respects. But of late, we've become especially interested in artificial intelligence and how it's impacting our world. We all know that AI is all anybody talks about, and we hear constantly about the possible dangers, the existential risk associated with AI, and all the areas where we need to feel some level of concern. It's in the area, of course, of scientific research and discovery that there seems to be a lot more upside than downside. And indeed, as we've heard over the course of some of these conferences, in the whole field of healthcare, broadly speaking, but especially in the area of drug discovery, there seems to be huge promise. And so having this discussion is a real opportunity for us to dive quite a bit deeper into both some of the kinds of scientific issues as well as the business issues that are part of what AI might be able to do to an enormously expensive and enormously time-intensive and an enormously difficult process made all the more urgent by both what we know must be done in terms of human health and what we believe might be done by yoking AI to some of the standard biological modes of both discovering new drugs and then, of course, testing them, doing trials, and rolling them out. But over to you, Seema.
Thank you.
Welcome.
Thank you, Nick. It's so great to have all of you here. And we both kind of gravitated towards AI because I don't know about you all, but from Davos and JP Morgan to TED and every meeting I go, there's 2 letters that keep standing out, and that is AI. There's all this hype, all this hope, and on the one hand, humanoids are going to take over Earth and control us all. We're going to lose jobs. And then on the other hand, it's all about the concerns, the fears, and inappropriate use of AI. But I think the fact of the matter is, I think many of you know AI, and especially in drug discovery and in business, is not a new flavor of the month. I mean, it has been used, and it is being used in the here and now and today. And today, what we're going to learn from our panelists really is how, on a day-to-day basis, AI is really accelerating and driving drug discovery today, and what are the ways that it's going to transform the healthcare landscape for tomorrow, and what do we need to be looking out for? How do we take advantage of this opportunity? How do we create the right kinds of guardrails and governance and, to take advantage, but also address some of the concerns that society has. So with that, I am really pleased to welcome, and I'm going to ask each of the panelists to introduce themselves as we get into this conversation. First up, we have, if technology works, George Church from Harvard University. George, can you hear me? Are you there?
Yeah, loud and clear. I hope you can hear me too.
Yes. Oh, there you are. Hi, George. Nice to have you with us. And I hope you can see us as well. We see you in 360-degree view. You are surrounding us all around the room on the video screen. So, your presence is well felt, as it should be. So thank you. Thank you for joining us. And then also here, I'm going to sit down now. George, can you say a few words about yourself? I mean, you need no introduction, but why not? Introduce yourself.
So I'm a professor at Harvard and MIT. I have worked on new technologies, reading and writing DNA and so forth. And apropos of this tonight's discussion, I've co-founded 5 of my 48 co-foundings are on AI, machine learning for drug discovery.
Thank you. Thank you, George. Thank you for joining us. And then to my left is Luisa Pineda, another great collaborator, founder and CEO of Envisagenics. I got to know Maria Luisa first when she and her little company at that time joined J Labs, which was one of the J&J Innovation J Labs right here in New York City. You want to introduce yourself?
Hi, I mean, I'm introducing myself after George Church, really? That's an honor. I mean, nice to meet you, by the way. Maria Luisa Pineda, I'm the co-founder and the CEO of Envisagenics. As Seema said, we spin out Envisagenics actually 10 years ago. So we're celebrating this Friday at a Cold Spring Harbor laboratory. I was a Watson or Cold Spring Harbor fellow there. And we spun out of Adrian Craner when we were developing the now FDA-approved Spinraza, which is an RNA therapeutic for children with spinal muscular atrophy. And the premise was using and utilizing sequencing, AI, machine learning, and RNA splicing to develop therapeutics. And again, there was no industry back then. And I had to figure out how to build something that was non-existent in an industry that was also non-existent back then. And J Labs was where we started our wet lab to the first time. And when we became J Labs, my actual mentor in J Paul, because that's why you get assigned, was Dr. Paul Stoffels, the vice chairman of Johnson & Johnson and chief scientific officer, who worked with Seema. So we got to work very closely every quarter. That's correct?
That's correct, yes. And you've come so far and such great things you're doing, and we're looking forward to learning more. So next to Maria Luisa is Grant Mitchell. Grant Mitchell is co-founder and CEO of EveryCure, along with David Feigenbaum, and the two of them co-founded EveryCure as a way of using AI to repurpose already marketed drugs for new indications. So do you want to introduce yourself? And you are a Cure resident and the winner of our Cure Exchange Innovation Challenge as well.
Yes, thank you, Seema. I once met George Clooney and I told him, I'm sorry, this is so much more memorable for you than it is, or for me than it is for you. So I'll say that to you also, George. Yeah, my name is Grant Mitchell. I'm CEO of EveryCure. My background, medical degree and a business degree from Penn and Wharton. And my roommate at the time actually is who inspired the genesis of our organization. He developed a rare disease that he nearly died from multiple times that started when we were in medical school. And we didn't have $1 billion or 10 years to develop a new drug, so we had to study the disease as it worked in his body and then test drugs that already existed on him, and ultimately found a drug that put him into remission. And that helped us understand the problem, but also the opportunity of drug repurposing, that there's these 3,000 life-saving precious molecules that we have of humanity, and they can do way more than they currently do. And so we use artificial intelligence to create the interconnectivity, the web, the matrix between drug and disease across all drugs and all diseases. And what makes us, a couple things that makes us unique is that we're agnostic to drug and disease, so we don't go after one particular disease or one particular molecule that we own. We let the data guide us towards what the best opportunities are. And we're also a nonprofit, so that we can be guided without a profit necessarily incentive.
That's great, thank you. And last but not least is Thomas Fuchs, and he is from the Icahn School right here at Mount Sinai. And tell us a little bit about yourself and how you use AI in digital health.
Of course, first of all, thank you for the invitation. It's great to be here and in this esteemed presence, which is, of course, fabulous. So I'm Thomas Fuchs. I'm leading the new AI department at Mount Sinai, and I'm the Dean for AI and Human Health there. And we try to apply AI across the field in all kinds of different areas. My own lab works on computational pathology. I have a PhD in machine learning from a time way before it was cool. And the world, of course, changed, which is great. A few years ago, we spun out a company from Sloan Kettering called PAGE, Pathology Artificial Intelligence Guidance Engine, where we use AI and build large foundation models for example, diagnostics, response prediction, and drug selection.
It's great. Well, thank you all for joining us. Let me start with a question that I'm going to pose to all of you, and then I'll come back with pointed questions to each of you, and then we'll open it up to the audience. So my opening question for each of you is, I often see AI referred to in the future tense, how AI is going to. And what we're here to discuss today is that AI we can be using in the present tense because it's happening here and now in the laboratory, in day-to-day work of that you are all doing. So why don't we start with this framing question, right? What are we doing right now with AI? And a revolution is being promised, right? So is a revolution coming? And what are you doing yourself or in your companies or labs to make that promise of AI in drug discovery real? George, do you want to lead us off?
Oh, okay, sure. So the ones that are closest to present tense rather than future are the ones that have already spun out of our academic lab, and those would be, and each of the 5 companies has something moving towards clinical trials. So this would be Jura, Nabla, Manifold, Dino, and Shape Therapeutics. And just as an example, and each of them use a combination of machine learning plus what we call multiplex libraries, where multiplex libraries alone don't work because you get very low yields without machine learning. And machine learning alone doesn't work because it's a kind of a poor simulator of complex biological systems like the whole body of a human or a test. But anyway, for example, we developed used cycles of AI machine learning plus these multiplex libraries of, say, 1 million-fold to inject primates, macaques, in with libraries that would allow drug delivery across the blood-brain barrier with expression in particular brain cells. And that would, we improved 100-fold on the previous best in terms of targeting these brain cells, and 10x detargeting of unwanted migration to the liver.
Yeah.
So that's an. I don't know if you wanted a longer than that. That's the short.
No. It was just like to have like real examples of the here and now, of what's happening and how AI is being used to accelerate drug discovery. So I think that example holds. And do you want to give an example from your?
Yeah, we can. I think what we're trying to do now is actually give people real proof of concept of what we're doing in each of our companies. So people stop calling us hype. Because in reality, I've been building my company for 10 years now, but it's accelerated. I'll give you like a dry lab and a wet lab example. The dry lab, when we started and we spun out of Cold Spring Harbor, first they used to charge me energy units because they used to have servers, and they charged me energy units to analyze a genome. And we used to do from a genome analysis perspective, around 8 to 10 weeks to analyze one genome. Right now at my company, on the cloud with high performance computing, with machine learning and all the AI and all the platform that we have built, we can analyze 2,000 full transcriptomes in under 2 hours. So 2,000 patients, full transcriptomes in under 2 hours. So that's what machine learning and AI and all these tech could really help out. If we didn't do that and engineered and work with Microsoft and Google and AWS over the years, plus work with machine learning, we wouldn't have been able to do this. That's just in the dry lab. On the wet lab, we've been developing therapeutics. So the way that we've approached it is by doing and building different models where we can kind of automate everything that the lab does. So for instance, on the RNA therapeutic and the antisense oligo, we used to do one nucleotide walks where we will design one nucleotide difference for each 16 or 18 mer compound through the whole genome. So then you will test 500, 1,000, 2,000 compounds per target. Okay, each compound is around $3,000 to $5,000, It takes time for you to test in cells and in mice, and then in human, imagine testing all those. Right now, we have the same machine learning algorithm, which we call splice learn, and basically it does that automatically. We only get, by doing a chromosome walk or nucleotide walk, you get 5 compounds that work. When we do our machine learning, we also get 5 compounds that work. Well, how? Because we've automated that process, and we understand that if we block a nucleotide, in a position, what RNA binding protein, what is the biology, the mechanism of action. So everything behind it is known by our scientists. So when we synthesize a molecule, 2 to 3 work, and we've just saved millions of dollars, unlimited amount of time, and then in a week we get them, we try them out, and 2 to 3 work. So if we didn't have this platform or these algorithms, we wouldn't be able to, either dry lab or wet lab do the work that we do at Envisagenics. So
that's interesting, and we'll come back to that because we're talking about cost, speed, efficiency, and I'll come back and we'll talk a little bit about can you actually achieve that kind of magic of getting everything right? Grant, what about you guys? How do you use AI like on a day-to-day basis? Give us a specific example.
Yeah, it's interesting. A lot of the, like George's company and your company, a lot of the AI, a lot of the applications are being utilized at the very beginning of the process, which is identify the target and design the ideal molecule in order to hit that target. Whereas every cure, we're dealing with drugs that already exist. They're already in the market. They're already safe and understood. And a lot of the mechanisms of action are relatively known. And so our task is figuring out what else can they do. And so when we use AI, we use it all along sort of the process of making this prediction on what else can this drug do. So it starts out with building a biomedical knowledge graph, which is this sort of 2D representation of all the biomedical concepts that exist. So drug, it's a target that's on a protein derived from a gene that's inside of a pathway and a cell type and an organ system that results in a phenotype. And so it's any database in the world, which there's around 180 that we're integrating right now, that connects a relationship between any of those concepts is a viable data set for us to integrate. And one thing we can do is we can use large language models to scrape all of medical literature to extract those relationships to construct that graph. Once that graph is built, we then use machine learning again in different models, whether it's graph transformers or other more traditional models to classify, to train the model. Okay, we have a known treats relationship. This drug treats this disease. Can we detect a similar pattern throughout the graph that's an unknown relationship, but is very promising based on the patterns that it's learned on? And then an additional validation processes after we find that hypothesis, whether it's using machine learning models in real-world evidence or protein-protein interactions to confirm what we suspect. So it's not just one model, one approach. It's really along the whole process of identifying and building a hypothesis and then validating it before we ultimately take it into clinical trials.
So, I'm just trying to think in my mind, if you had to hire a bunch of postdocs to do this job, how long would it take and how many man hours, women hours, people hours would you need?
Well, I mean, that's especially for what we're doing, which is this massively parallel thing, which is all 3,000 drugs and all 22,000 human diseases, cross-referencing, making a normalized score from 0 to 1 of the probability of that drug to work for that disease. So 66 million possibilities. To do that with humans would just literally be impossible.
Yeah, the computational power, you've just kind of like, it's just gone beyond control. Yes. Thomas, what about you? What are you working on today in your labs that you can give a specific example for?
Yeah, so of course, AI only came into the public consciousness thanks to ChatGPT, but of course, it was applied especially in the imaging space and also, of course, in the drug development space much earlier. So What we are working on is we use digitized images of pathology samples, for example, a breast biopsy, prostate biopsy, and pathology really has not changed in 150 years. They literally do the same thing in 90% of places in the US as they did 150 years ago when Rudolf Virchow invented it more or less in Berlin. So it's very subjective, but it's of course the critical point because you don't have cancer until a pathologist says so. So it depends on the pathologist, what kind of treatment you get, what drug you get, and so forth. So AI can help on many levels here, of course, to make it more objective and also more personalized. We built these large machine learning models to not only detect cancer, classify it, but also, again, predict sometimes underlying morphological changes, underlying genomic changes that lead to morphological changes.
Yeah.
In 2021, we got the first breakthrough designation for an AI in all of oncology. And 2 years later, that led to the first approval of an AI in pathology. And it's still the only one. While you have hundreds in radiology, you still only have one in pathology. Because the FDA treats diagnostics differently than radiology, which mostly is, of course, screening. You can come back to that. And that's now used on 4 continents across the globe. We had 10,000 patients diagnosed last year alone, 2023, with the help of the AI. So it is here. If you have a digital pathology lab, you can use it. The problem is, of course, that conversion from 150-year-old discipline to digital takes quite some time. And these days, we build much larger foundation models from millions of these slides, billions of images. So the last one, for example, it's named after Virchow, we used 10 times more data than all of Netflix. So think of all the shows you watch, all the movies, all the episodes, every single pixel. That's just 10% of the pathology data we pipe through.
Wow.
And at that level, you now have a frontier model that understands morphology of cancer and normal tissue at a very meticulous level. And you can use it for CDXs, for biomarkers, for trial enrollment, for example. And then, of course, diagnostics far beyond prostate and breast, but also for rare diseases.
How widely used is it? I mean, because, you would think that something like this could lead to huge transformation in the diagnosis, treatment of cancer. Is it already having an impact, or is there a time lag? How do you make it widely used, and is it accessible?
Yeah, so it certainly will. So one indication of that is, so we coined the term computational pathology 16 years ago. And of course, nobody cared, because nobody cared about machine learning and nobody cared about pathology. And these days, and you probably saw it at J&J, every big pharma company has their own computational pathology groups internally, because it will be an integral part for not only diagnostics, but again, fast identification of patients to know who gets the right drug. So the impact is completely clear. It's certainly hindered again by the digitization of the whole field, which is slow. And of course, all hospitals are in red after the pandemic, and that holds it back. There are many European countries who are far advanced because there are governmental incentives actually to use new technologies. That's unfortunately not so much the case here, as you all know. And the US is behind in that area for sure.
US is behind in that area. George,
in clinical adoption.
In clinical adoption, yes.
Forefront of
not in the forefront of development, the clinical adoption. Okay, well, we'll come back to that in a second. But George, I want to ask you a question, which is, we have genomic data, we have EHR data, we have data from so many different sources, pathology and, radiology. How do you integrate all of that data and make sure that you're understanding the body as a whole, and how can AI be used in that way? Or is it too far field ahead for what we're discussing about the here and now today?
Well, I gave my best example of the here and now, which is protein design, drug discovery. But what you ask about, I think has an interesting limitation, which is that there are certainly a lot of data, but very little of it's integrated. You have very few examples where you have a person with all of the medical records and genomics and omics that you might expect in a clinic today or in the near future, available as an integrated unit. So usually it's spread out where you'll say, this is the allele frequency in this population, cases and controls. And you won't have all the other genotype, phenotype, environmental data that you need. And this is one of the things we set out to do as a demonstration of the Personal Genome Project. And I think that would be extremely valuable to take advantage of this exponential improvement we have now in machine learning. I think that would be a great combination.
Since we did talk about protein folding, can I follow up with a question about AlphaFold and AlphaFold 3? And I mean, it's able to do in fast fashion the work of God knows how many postdocs in terms of protein folding. Years of work done in a matter of seconds. What do you think about that, and how does it impact the day-to-day work of graduate students, postdoc, people who are working on projects in, for example, your own laboratory?
If we can, it's hard to talk about where AlphaFold and other tools are going. So I'll talk about where they've been. When AlphaFold be compared to large language models focused on the sequence rather than the 3D structure, AlphaFold does satisfy the 50-year holy grail search for folding, but it can't. The fold can't tell you the function that easily. So, for example, if you substitute a serine for an alanine in the middle of a serine protease, it will fold to high precision, but it won't be catalytic. And so you either need the large language model without the 3D structure, or maybe put the two together. But it really truly both categories, the protein design that's focused on function and the protein structure, which you can, I think increasingly has been the necessary for finding docking of small molecules and so forth. I think it's just an amazing time.
Right. Thank you. So, bringing it back to the patient, right? Some of the work that you're doing is either already having or can have a very immediate impact on patients, right? In terms of cancer, in terms of like the immune system. Talk a little bit about that and how this, so because we're talking about drug discovery, but ultimately our goal is to find cures.
Yes.
And, treatments for patients. So tell a little bit about that.
Okay, so at Envisagenics, what we're really trying to kind of like use machine learning for was, we've developed this platform, we use grants, we use funding, and we have these amazing partners, but we kept getting, a lot of interest on, protein structures. We are an RNA splicing company, and RNA splicing, a lot of people don't touch it because it's very complex. It's very, people don't really, understand it that well because there's not that many experts in the field. But then when we design and build a platform to better identify, targets and proteins, we realize a lot of the RNA splicing protein isoforms were built by these, RNA errors. So by doing that, we actually end up building these humongous, I call it a database, but it's a functional transcriptome reference that has, I'm saying it correctly so my co-founder doesn't hate me, because the reality is, for me, it's a database. It has around 14 million possible or potential drugs for patients for different cancers. But the premise is, which one of the 14 million is an actual, can be used to develop a therapeutic, an actual drug? So that's where AI and machine learning comes from. I call it filtering, or, kind of like going through it, the 14 million, and start narrowing it down. But depending on the modality, we focus at Envisagenics to develop immunotherapies. So identifying proteins that are in the extracellular membrane of these tumors from patients that are not present in normal tissues, that we can then basically identify them, are very specific in tumors, have like a little flag, and that flag could then be, with your immune system built into a, conjugated drug or a CAR T. So the immune system can then go and eat it up, and the tumors could be gone, so the patients can be cured. So that's called immunotherapy. And the premise was, hey, identify these proteins that are based on RNA splicing errors, and then that are tumor specific. Use AI to go from 14 million to maybe 5. That you can then design the antibodies, get immunotherapies, and then in a year take them to the patient. So right now, that's how long it takes us.
Where are you?
Where, once we do all the R&D, our biopharma partners then take them into the clinic. And yeah, in 12 months, now from starting a program, analyzing RNA sequencing, to all the way to a antibody that could then be humanized into a immunotherapy, it takes us 11 to 12 months.
Wait, that's like, that's fast.
It only took us 10 years to build the platform, but yes.
Because I know we've always heard that it takes, 10 years and $1 billion,
Yes, but I think that is the beauty of using this technology to really advance drug discovery, and it's here to stay.
It's great. Well, thank you. Grant, in your case, right, you already sort of figured out a use case with David in some way, shape, or form. Not for the AI piece, but for you figuring out, new indications for existing drugs. With AI, are you still in the exploratory stage, or have you actually hit upon a few areas where you think you can take things, forward faster?
Yeah, we do have a number of leading sort of concepts and ideas that we're assessing, and we've also sort of spot checked the accuracy of our predictions in a few diseases that we do know well, such as Castleman disease, and even saw a patient move out of hospice when they were told that nothing else is going to work, and the number one drug that we had predicted for that disease was tried for that patient and led to them, recovering. So what's exciting to me about drug repurposing is that it closes this feedback loop for validation of these AI-driven predictions. And so I'd be very curious about, for the for-profit companies today, how they're thinking about that, because not only do you have to support an AI platform to generate the predictions, is this drug going to work for this disease? Once you've made that, you now have to go through the valley of death that every other biotech goes through, whether they're an AI company or not. And in the meantime, you have to continue to support your AI function that doesn't know if they created something of value or not. And so there's a tension between these two entities that are normally not inside of one company. And so with right, yeah. So with with drug repurposing, we can sometimes go immediately into patients. Like in Dave, my co-founder, we went in 2 weeks after we had the idea because he was in a desperate situation. Yeah. And then, whether it works or not works, that feedbacks into the platform to validate. But I'd be curious for the for-profit folks, what I've seen is that some of these companies are struggling because the market values the asset once it's getting into the clinical stage. Yeah. And then they don't know how to value the AI component that got it there. So I see the CapEx all of a sudden shift dramatically from the platform build into okay, let's just make sure this gets through successful phase 3.
You're right. The platform does not get any value. The bioinformatics, they used to call me a bioinformatics company. Zero value.
Yeah.
Okay.
The second it gets into the clinic, now you have to change.
But yeah, I agree with you.
You have to call yourself a cancer company or an immunotherapeutic company.
A therapeutic. Mm-hmm. But I agree with you. All of us, at least in AI and drug discovery, which now the industry has a name. The industry didn't even have a name. Sometimes they used to put with like Apple Watches. Like a healthcare IT.
Hey, yeah.
Right? Mm-hmm. So, it's changed quite a lot, but I agree with you. All of us are focused on product, and the product is a therapeutic.
Mm-hmm.
But Thomas, your model is slightly different.
Yes, we also don't do drug development. It's really more finding the right patients for them.
So you focused a lot on the platform.
Exactly, yes.
right? Yeah.
So there it's clearly, so first of all, there's a clinical platform that's used in Brazil, in Sweden, in all kinds of places in the US. And it really helps pathologists to just sign out digitally. It can be remote. You have all the slides, you have all the information. It's linked into the LIS and so forth. But on top of that, you have the AI that can help with many different tasks. So the standard sign-out process, just make sure that you actually find all the cancer. So one of the applications the breast pathologists love most is actually for lymph node assessment, because every good pathologist, when they go home, are enormously anxious that they missed something. Out of these millions and millions of cells on a single slide, it just takes 2 or 3 to completely change the treatment path if your nodes are positive, right? And so to have that affirmation that you really saw everything and didn't miss anything because the AI is highly sensitive is a great asset for the patients and also, of course, for the pathologists. Then what's more interesting, for example, for various targets is we have approximately 120,000 sequenced patients where we have the match between sequencing plus so somatic and germline sequencing plus the slides. And then you can develop actually AI systems that from the morphology of the slide could predict, for example, if the patient is EGFR positive, FGFR positive, or for example, MSI, something that's rather straightforward, and then more difficult things. And if you can do that from a $5 slide instead of waiting weeks for expensive sequencing, then of course you can also think of really democratizing access to healthcare. And that's in the rural areas here in the US or even globally where you would never be able to afford sequencing. Or from a pharma perspective, if you have a budget of, I don't know, 1,000 patients you can sequence, and you can actually pre-select based on the histology, and your yield is much higher, you then suddenly get twice as many, of course, with I don't know, if they're red positive or whatever you're interested in. That, of course, does only work for driver mutations that actually lead to a morphological change, which is not true for most of them, but for some it is, and that can be really a benefit. Even if you think of EGFR, where you have a targeted therapy, it's one of the oldest ever. Even in the US, between 30 and 40 lung cancer patients don't get tested. Right? And if you get that at point of care immediately, so the lights go off and say, look, for this patient, maybe they could fall into this trial, that would be enormous benefit added, as long as you're digital.
Just incredible, and it's it's like a little disappointing to hear that the adoption here in the United States is so low. George, you've been involved in dozens and dozens of innovation, and I think you said some number at the very beginning, at the top of this, like 48 out of however many companies involved in AI and machine learning. I mean, how has AI really changed the landscape of innovation and company formation? Coming out of your labs, has it changed and a lot? And is it going to change more as students and other innovators start to think about how they do drug discovery and how they spin out companies using AI?
Well, it's 5 out of 48 that are dedicated to machine learning, but I think it's very quickly transitioning to it's just standard practice. It used to be very special to be a biochemist rather than a chemist or a biologist. Now, basically all the biotech companies do biochemistry. They all do computational biology. They all do genetics in some form or another. So, I think it's getting to be ordinary. Yeah. Interestingly.
Yeah, and it's here to stay. It's like a computer. It's all like an iPhone that everybody uses now, and so it's standard practice.
Or a calculator.
Yeah, I mean, like a calculator, right? Yeah, I mean, with ChatGPT and LLMs,
It's like the internet.
It's like the internet. It's part of the DNA, right? But what do you predict and what do you expect to see in the next few years? Let's say 5 years, 10 years.
Well, I think that there's going to be a big increase in this, what I call natural computing, which is combining these amazing things you can do with silicon, von Neumann digital, and actually synthesizing perfect simulators, that were, and very big libraries. So now we're getting in the order of 10 trillion members of a library. If you can do an in vitro assay, it's far more accurate than anything you could do in silico. But it's all in silico designed. So I think we're going to see a lot of that going forward, especially in biology and medicine, where you can actually make 10 trillion things cheaply. We're not going to be seeing the same kind of effect, machine learning on, say, astrophysics. We can't do the experiments. We just have to take in the data. But it's clearly very data hungry, and this is a field where you can give it lots of data and go through loops of analysis and synthesis.
And speaking of data, it's at the end of the day, machine learning, AI is as good as the data you put into it, right? Garbage in, garbage out, or if it's got real data. And so there is concerns around that,
Yes.
right? And concerns about the accuracy and the validity and the integrity of the data, the completeness of the data, right? If you have incomplete data, what are you getting as a readout? And then in terms of diversity and equity and inclusion and all of that, right? What you have in your database we've been talking a lot about women's health recently here. And so, if you have all the data based on men, then how are you going to cull the data to apply to women? So there's lots of different still questions, right, about data and how you use it, and
I think that was going back to George's point, where the government probably will have to, kind of get involved. And probably Thomas was saying the same thing, where we do need, as an industry, but it has to be a combined, government and corporate, and not a single company could do it alone. Not the Arches or the flagships or any George's company, which are really well funded. But despite that, you can never get unless you ask the right question and maybe design and get the data, that you need to answer a specific question using a specific algorithm, then you could, curate your data yourself, find the right patients, right? But, even my data scientists and everybody I speak to, the data you get is the data you get. I wish we could get better data, yeah, but then you have to make it yourself, or find the right partner,
data sharing,
or negotiate for it, or pay for it. There's so many issues with it. So I think government, corporate, I think it has to be a big consortium that really has to do with it. Yeah. DNI, right? Even the algorithms. Who writes the algorithms? A lot of my tech friends used to say, when they were testing the face recognition for TSA for a long time, they started realizing that all the algos were developed by Caucasian engineers. So when they were testing it, they didn't recognize anybody with color because they didn't even realize to put it into the algo. So, little things like that, right? Where, not only the data input, but who develops the algorithm and their background. So for biologists, for data scientists, it really depends on the question you're asking and how you're feeding that data. But it's in curating the data, trying to get diverse, at least for Envisagenics. My co-founder's Argentinian, Israeli. I'm Colombian American. So I try to do a lot of foundation, work with foundations, so we could work with the foundations directly and get their data input as strategics, with grants, with all these things. My co-founder, it's setting up Israeli connections with partnerships, Middle Eastern, some in Latin America, because we need diversity, some of our Asian partners as well, in Singapore, right? So it's how do we get all these data from different people? Because diseases are not, are global.
Yep.
They happen to absolutely every single person, so it doesn't matter where you're from or your nationality. So I think that's where, government, And somebody like, the Clinton Foundation, right, like in human and women's health are really pushing the envelope forward.
Yeah, absolutely. Grant, there's this golden thing, right? Ratio of speed, cost, and accuracy. We've always been taught that you can only get 2 out of 3. What are we thinking about with AI? Cost and speed, what happens to accuracy?
Yeah, so, I guess, is that me? No? So when I used to work for Quantum Black, which is McKinsey's machine learning arm, and whenever we would tackle problems for clients with machine learning applications, it's basically, can it be better, can it be faster, can it be cheaper? And some say you can't make something, all 3 of those, and at least in the healthcare services space. But I think you can in drug development. You can design a better molecule. You can do it faster, as you described, and the whole process of doing it better and faster makes it cheaper. So I think AI absolutely unlocks that sort of panacea of better on all fronts. But at the point earlier we were talking about data aggregation. That's a problem that actually Evercure is trying to solve, and it's why we're a nonprofit. Because we want to be collaborative, we want to be non-competitive, we want to be a place that's the Switzerland of data that people feel comfortable sharing, so that it both contributes to the field, but also benefits them because they get a better model back that we can share with them. So, as a nonprofit, if you want to build the best ad platform, we figure, well, we need the best data set, and the only way to get it is to be collaborative with people, and that allows us to go after drugs that we don't own. And it allows us to go after diseases that the pharmaceutical industry neglects, and populations that get neglected. So hopefully we're able to crack that nut with the organizational structure that we've designed to address a lot of the issues you're discussing.
And how do you think about working with governments, to sort of standardize things? Because clearly it's not yet there.
Yes, I mean, there are 2 parts of it. One is, of course, the regulatory side, and the other one is then reimbursement that's sometimes driven, of course, from the government. So regulation is very important, and you actually spoke to diversity, and you brought that face recognition example, right? So that these days would not have ever passed a FDA approval if it actually would put it in front of the FDA, because proving out that an algorithm is equitable and really works for everyone, at least for the AI algorithms now, is part of that approval process. That's why that is an important step. Right? You might even not understand how your algorithm works, like you don't understand how, for example, propofol works, but you have to prove out that it's safe, effective, and equitable, that it really works for everyone. Mount Sinai is in a very lucky position because it's the largest health system in New York City, 8 hospitals, 400 outpatient locations through Queens, the Bronx, and everywhere. And these are the most diverse areas in the world. So you actually have data across many, many backgrounds. And that's absolutely key if you build AI to prove out that it works for everyone. But then on the regulatory side, for example, in pathology, there were many loopholes that still exist, and that's were used by sequencing companies, by AI companies. On the imaging side, these are lab-developed tests, where you actually don't have to go through studies and the FDA process. It was just recently that the FDA announced they're going to close that loophole. That's a big change. Mm-hmm. But I think complex systems like AI that can't be understood by a single lab director have to be regulated at that level. And that, of course, also has the benefit that it really differentiates the wheat from the chaff, because we live in a world where there's enormous hype, enormous fluff, right? There are, 15,000 biotech companies, right? Half of them, of course, have never actually proven on many diverse population patients if the algorithms actually work. So regulation is important in that space, also to give then the assurance and the security to the patients. And it's regulation is actually not the main hurdle, right? The main hurdle is reimbursement at the end of the
Yes.
day. And that's, of course, very healthcare system dependent. And as we all know, right, we are in the AI healthcare winter, right? It's a bloodshed in the streets. So the startups are dying by the dozens every single day because you can build a new LLM company every single day, and Deerfield will give you 100 million, I guess. But if you actually do AI in healthcare, nobody gets anything these days, right? Because there is just, it's too hard. Right, consumer language models, drug development are the more lower-hanging fruits compared to healthcare, and that will take some time to actually break into it. So that actually goes back to your first questions. There are many tools available that provably can, for example, save lives. Literally, the FDA stated that, for example, that are not used just because of reimbursement questions, of the slowness of the healthcare system here. And that actually holds everybody back and is to the disadvantage of patients. So patients are for sure dying today, not because of the AI and Terminator fears, but because of the lack of AI. And I think we really have to change that.
George, I think we're reaching the end, but I want to give you the last word. Any words of wisdom for all of us here? What should we expect in the future? What should we watch out for? Reason to believe and hope.
Wow. Geez. Well, I think one thing, David Baker and I just wrote a piece on things to watch out for on the negative end of the spectrum, and things we can do to mitigate that. On the positive end, I think we're going to start seeing AI creeping into the technology development. So a lot of this is limited or becomes unlimited. The exponential is tracking the technology, the instruments, the different ways of multiplexing and different chemistries. When you get that autocatalytic self-fueling cycle where the AI is inventing new technologies, which can then feed it more and better data, less garbage in, then you're going to see a true explosion.
It's great. Well, we can't wait. Well, thank you. Thank you for joining us, George, and thank you to the panelists, Maria Luisa, Grant, Thomas. Thank you for making time. It's been a great conversation. Nick, thank you once again for the partnership. Appreciate it. Bye all.